This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 1033, recorded on August 4th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 84, mostly sunny, and that's probably around 29 degrees Celsius.
Here it's cloudy and 26. Also joining us from Montreal, Canada, Angela Mingarelli.
Hello. How's everyone doing? Here it is 75. So that's, I think, 24 Celsius. It's partially cloudy. It's pretty nice, actually. I just took my dog for a walk.
What's the dog's name?
Odin.
Oh, that's one of the Norse gods, right?
Yes, exactly.
Very good. That's cool. Odin, great name.
Mm-hmm.
What was the characteristic of Odin? Do you remember? Was there something?
He's the god of war and death.
War and death. A good name for a dog.
But he's only like 18 pounds, so it's kind of funny. But
what kind of dog is it?
He's a mixed breed. I got him when he was like 10 days old when I was living in Spain. I had no idea what type of breed he was. I basically saved him. I rescued him. So
was it hard to get him in the U.S. from Spain?
Or Canada, for that matter.
Yeah, actually for Canada, it's okay. Yeah, in Canada, all you really need is a rabies vaccine. So he had to be vaccinated and then wait minimum of 3 months after vaccination to enter. So it's pretty easy. Pretty standard. You just
brought him on the plane in a carrier, right?
No, he had to go, well, yes, in a carrier, but under, not with me.
Oh, he went in a cargo.
Yeah, unfortunately.
Must be freaky to be down there. Must be noisy and stuff, right?
Yeah, they have it temperature controlled and pressurized as well, but you can only travel with animals. I think it's November to March, or no, November to April. You can't. So it's from April to October you can because of it's too cold under the plane.
But at 38,000 feet, it's always cold, isn't it?
I think it's more so the transit in the airport and going through the plane, the cargo, like the loading and unloading. Yeah.
I heard this story once of a lady who brought her tiny dog onto the plane. It was one of the companion dogs that you could bring on. And this flight attendant made her put the dog in the overhead compartment and it died.
What?
Can you imagine?
Well,
I mean, she was wrong. She was wrong. But there's can you imagine at the end of the trip? And I just can't imagine how horrible that would be.
That's terrible. And that's actually illegal. Like, you're not supposed to have any live animals in the overhead compartment. They should always go in front of you, in front of your seat, like your luggage. They should just go there.
Yeah.
The plane I was on day before yesterday, there was a dog across the aisle under the seat in front. Mm-hmm. And their paw was sticking out in the aisle as people were. And I pointed out to the owner, I'm worried about that paw. So
I was on a flight where this young lady had a very large dog, lovely dog, but really big. She was in first class. When I walked by, the dog was right in front of her, took up the whole space in front, and her neighbor was kind of spilling over into the next person, and he was okay about it. He said, "It's okay. I like dogs." But can you imagine? Imagine a cranky person. I don't want to sit here.
Oh, yeah.
All right. Sorry about that, folks, but this is what we do when we see each other after a while. We just chat. And I'm sorry that you have to be subjected to it, but you can always fast forward. Today,
everyone loves dogs. People talking about dogs.
I don't know if everyone does. I don't know if everyone does, but many people do. I think they're wonderful, and when I pass them, I always look at them. This morning, a young lady had two young little dogs on a leash, the kind that are really close to the ground, right, with longish hair, right?
Dachshund. Like
not a dachshund. No, not a dachshund, but a small dog with very long hair, black. Anyway, she stepped off the street. She was in the street, right? And cars were turning and they can't see the dogs. I'm like, dogs are going to get run over. She wasn't paying much attention. I felt badly for them. Anyway, Today's the birthday of Altina Shinasi. She would have been 116, but she's not around anymore. But she designed the cat eye glass frames, which I love, which are just great, right? Which your third grade teacher wore. It's so cool. And it's on the Google, what do you call the thing at the top of the page, right?
The homepage, yeah.
Homepage. And there's a picture of the glasses with her inside one of them wearing it. It's just a great image.
They're cool.
So happy birthday.
It's also my grandmother's 99th birthday today.
Wow.
Yeah.
Wow. And she's
That's
still
some
around. She's
longevity
still
genes.
around, right?
Yeah. She's this is my Italian grandmother, my father's mom. So Signora Mingarelli is 99 today.
Where is she living?
She's in a residence actually because she has dementia. Like she's physically pretty good, but yeah, she has dementia, so she lives in a residence.
In Spain or in Italy or?
No, here in Montreal.
Is that her house? I think it's her house that you're in, right?
Yes, exactly. I live in her house. She doesn't live here anymore, but I saw her a few days ago, actually. Went to see
visit?
her.
That's nice.
Mm-hmm.
Cool.
Yeah.
Well, happy birthday, Signora Mingarelli. Okay, so we have a letter from Susana Lopez from a university in Cuernavaca, Mexico, who's a friend of all of us. Given the warm weather, to be kind, and rain we're having in most of the northern hemisphere, the mosquitoes are in their best shape, taking advantage of the great public that you have in This Week in Virology. I want to remind you, we made a book for children entitled Paul and the Mosquitoes That Could Be Helpful These Days. The book is online free of charge. It's in Spanish, English, French, German, Romanian, and maybe you can suggest it in one of your programs. And we'll put a link to a site where you can download them all in various languages. And she writes, I think it's a good information to spread this summer. And so, yes, they're great books written by Susanna and her colleagues, nicely illustrated in multiple languages. So,
yeah, I read through the mosquito one again. I enjoyed it.
Yes, the mosquito one is cool.
It's really good pictures.
Yep,
really.
Pictures are great. Yeah, there are a couple of them now, besides the mosquitoes.
Yes.
And we also have Amy Rosenfeld at the FDA is looking for a second lab assistant. So she hired one person through TWiV, who is arriving shortly, I believe. And now she would like a second. So we'll put a link to that job description in the show notes. It is research assistant in CBER, which is part of FDA, to work on enteroviruses. And Kathy, you have a letter from Mary Woolley.
Right. So Vincent and I have both plugged Research America several times and Mary Woolley, and this organization has great advocacy for research in America, as you might expect from the title. And I just wanted to tell you what was in today's letter. A Gallup poll released just last month shows Americans continue to lose confidence in major institutions, including our medical system. Confidence in the U.S. medical system has dropped by 10%, from 44% to 34%, a larger drop than any other institution except the presidency. Small business commands the highest level of confidence. And then she goes on to say, confidence improves when the questions ask about individuals than institutions in the surveys. Our January 2023 National Public Opinion Survey indicates that scientists, healthcare providers, and public health officials remain trusted sources of health information. So that's good news. We've long maintained that public engagement by individual members of the science and health community is an important part of the answer to bolstering public confidence in institutions. It's time to place a higher priority on assuming that members of these valued professions are empowered to engage the public. And Research America is working on ways to do this. But as Vincent points out, we are working on this by doing TWiV and hopefully empowering you in the science and health community, those of you who have listeners who fit that criteria, to feel empowered to engage the public about things that you know about science. So, end of PSA.
I would like Research America to spread the word about people who are doing this, not just us, but other people who are doing science communication. It would be nice. I know they have a lot on their plate, but they have a pretty broad reach, so it would be cool. That's where I got the line, when someone asks what you do, you tell them, I work for you. That's what Mary Woolley gave in one talk I heard years ago, and I love repeating that. I wanted to bring your attention to an article in Nature. It's not paywalled, so you can read it. Dengue is breaking records in the Americas. What's behind the surge? 3 million cases of dengue have been reported in the Americas so far. That's makes this year the second highest annual incidence since 1980, when the Pan American Health Organization actually began to track the number of cases. And there's a wonderful graph. This is why I want you to look at this, showing the number of cases just going up and up and up. And, we don't know why, but it may be that increasing temperatures are part of it, that have longer mosquito seasons and so forth. But they
spoke about how El Nino could maybe be influencing these spikes, but I wonder if this 2016 spike also had to do with that, because it seems like the previous is that 2016-ish, I guess. There was a really high spike. I wonder what was that? Yeah. Almost similar to this year.
There's another one in 2019. Was 2016 a Nino year?
I'm gonna check.
Okay. Researchers at the University of Michigan in Ann Arbor have investigated how rising temperatures might affect it.
Right.
Do you know computational epidemiologist Andrew Brower? Yeah.
Don't know them. No. And they also talk about maybe longer seasons that's coming with global warming may increase the dengue season. So
2019 was an El Nino year, so maybe that could be, because it's once every few years, right? So maybe it is influenza.
I think so. I don't know if it's actually a fixed period, but if you look at it, the high years seem to be in multiples of 3.
Yeah.
Kind of. Not so much as you go further back in time, but yeah.
So anyway, check it out. Now, what could you do? Well, vaccines would be useful, but as they say in the last sentence, there are two vaccines approved, but they have not been widely adopted due to issues with efficacy, safety concerns, and high prices. So,
right,
unfortunate. But there's another one in the pipeline that maybe has fewer safety concerns that should be coming out soon. So we'll let you know about that. And also, we've had another passing in the virology community. Last couple of weeks, Mike Oldstone passed away, and now Harald zur Hausen. What was the date? I don't see it here. This article in Nature is 21st July. I don't know when he passed away, but recently.
Yeah.
But anyway, Harald zur Hausen proved that human papillomaviruses cause cervical cancer, got a Nobel Prize for that in 2008. He died at 87.
No, he didn't die in 87.
No, at the age of 87.
Oh, thank you.
Yeah, no problem.
I was still trying to figure out the year. Sorry.
It was in this year, I assume in 2023, right? But I don't remember exactly when. Anyway, I interviewed him a couple of years ago. Let's see, we can search for Racaniello, zur Hausen. He was lovely to interview. Yeah, Microbe World Video, episode 80. Where can you find this? It's on YouTube.
He died May 29th, 2023.
Okay.
I just found that as well in Wikipedia.
I'm going to copy the link and put it in. So I interviewed him in Manchester, England. It was the Society for General Microbiology meeting. And we were just standing in like an atrium with people moving around. So there was a lot of background activity. It was kind of cool. And we were just talking to each other. And the video is very nice. It was shot by Chris Condian and Ray Ortega, who both worked at TWiV 1000. I gave them a reunion because they used to work together for ASM, and then Chris left ASM, and now Ray remains at ASM. So at that time, 2013, I think, they were both working together. So check that out. It's a lot of fun. All right. We have a snippet for you, and this is something that Angela picked. So I'll hand the reins to you, Angela, and tell us all about it. And you know what's funny? You picked it on your own, and I picked another paper, and they're both science immunology papers.
Yeah, I was just about to say that. Yeah. And they're both, unfortunately, behind a paywall. I'm sorry for the viewers. I'm sorry. So the snippet that I chose is Science Immunology, as Vincent just said. And the title is Modulation of Type 1 Interferon Responses Potently Inhibits SARS-CoV-2 Replication and Inflammation in Rhesus Macaques. So there are 3 co-first authors. I'm going to try to say the names the best I can. Elise Voix, Timothy Huang, and Amit Udapaye. And there are 2 corresponding authors, Stephen Bossinger and Mirko Piardini. Is that Piardini? And they are from the Emory Vaccine Center at Emory University. And I kind of liked in their introduction, they said something that a lot of people maybe don't downplay, but some people try to downplay the severity sometimes of COVID-19. And I think it's important to acknowledge that there have been almost 7 million cumulative deaths due to this virus, and that it's important to continue research efforts like this research group that we're going to talk about their work to better understand this virus and ultimately save more people's lives. Because you think about that, 7 million people is a lot.
People always downplay it on our live stream.
Yeah.
People are some people sneak in and they say, "Oh, this is nothing. This has been nothing. This virus." I mean, come on, 7 million. It's not right to say it's nothing.
Exactly. I just wanted to, I think that it's important to put that number out there and for people to think about how many people that actually is. So this paper sets out to better understand the ways in which type 1 interferons regulate SARS-CoV-2 replication and disease progression, because these are still incompletely understood. So I know that we talk a lot about, or we have spoken a lot about type 1 interferons in the past on TWiV, but because we're trying to explain everything the best we can, I'm going to do like a quick review on type 1 interferons. We know that they play a central role in innate antiviral immunity, meaning that they protect us in acute viral infections. The interferon 1 receptors, which are the IFNAR receptors, are universally expressed on cells, and the activation of these receptors trigger downstream transcription of interferon-stimulated genes that mediate antiviral effector functions, and they also recruit inflammatory cells. And we also know that this is important and plays a role in COVID-19, so in SARS-CoV-2 infections.
In fact, we did a paper, I don't know if you were on, we showed that people with autoantibodies to interferons have severe COVID, right?
Yeah, I wasn't on that. I heard the TWiV, but I wasn't physically on the TWiV.
And then last week,
really interesting.
40% of West Nile encephalitis patients have autoantibodies to interferon. So very interesting development, yeah.
Yeah. So they used an approach using a non-human primate model, the rhesus macaque, which is a model of mild to moderate SARS-CoV-2 infection, to dissect these roles of antiviral and pro-inflammatory interferon responses early in SARS-CoV-2 infection. And they did so using a mutated interferon alpha-2, which is an interferon modulator, which has actually shown to reduce the binding and signaling of endogenous type 1 interferons. So,
and these macaques, by the way, they don't get severe disease, right? They just get mild disease.
Mild to moderate disease, exactly. Yeah, they have lung pathology, and but they don't have severe respiratory distress or death. As far as I can see, none of them died. They didn't speak about. It's not like comparable to humans necessarily, but it is comparable, like I said, to a mild to moderate SARS-CoV-2 infection. And because this paper has 8 figures, we're going to just like quickly summarize the findings. And
did you mention the authors or where they came from? I missed hearing it if you did. Sorry.
Yeah, they're from the Emory Vaccine Center. They're 3 co-first, 2 corresponding. Yeah.
Okay.
So they basically, so there's, like I said, there's 8 figures. The first 2 figures, they administer this interferon modulator to uninfected rhesus macaques, and then also to Calu3 cells, which are human lung cells. And they determine this, the impact on the ISGs, or the interferon stimulated genes. And they see that there is a slight upcrease in these ISGs, but none of the pro-inflammatory cytokines that you would see upon infection. And then they determined that in the Calu3 cells, they infect them with SARS-CoV-2, and that this interferon modulator can control replication levels similar to, what is it called? Normotrelvir, which is one of the components of Paxlovid, with, I believe it is ritonavir.
Just remember normotrelvir. That's what the Pfizer people told us when we were doing the podcast. They just say normotrelvir. Even though you see it written, it doesn't look like that. It's weird, and it's a fictitious name too. So
yeah. So then they demonstrated as well. So they saw that in vitro in these Calu3 cells that there was controlled. That inhibited, the replication of SARS-CoV-2, comparable to normotrelvir. And then they demonstrated that this is in vivo in the macaques, that this interferon modulator also reduced viral loads, it reduced lung pathology and inflammatory cytokines and chemokines in these infected macaques with SARS-CoV-2.
The timing is they're giving it before infection, right?
Yeah, so they do it actually, in this case specifically, they give it before, like -1 days. They also give it on day 0, and then they also give it throughout the paper, like on days 2 or 3.
Continuously, okay, yeah.
Yeah. And, sorry.
It's interesting that this it's an altered interferon of some kind, right? That it just so happens to not induce the inflammatory cytokines, but it's okay at some of the antiviral ISGs, right? I don't know the exact structure of this IFN mod. Do you know offhand?
They have it they explain it in figure 1, I believe. They show, was it figure 1? Oh, no, they don't actually. I thought they did. No, they just say that it's an interferon alpha 2 mutated interferon alpha 2.
So it's a protein. It's an altered protein that they give to the non-human, to the macaques, right? To
the macaques, yeah.
They inject it. How is it given to them? I forgot.
It's intramuscular.
So it's a protein that they're given. Okay.
Yeah.
So I think you were saying that when they give it to the macaques, it limits replication, right? Just like it does in the cells. Correct?
Exactly. It limits replication. And then they also see that it limits lung pathology. It limits cytokine production in the lungs.
Right.
And viral loads overall as well in the macaques.
Another way, by inhibiting the interferon, this is what happens. So in other words, the presence of interferon would make all those things happen that it's
increased.
Inhibiting.
Yeah. Right. Well, not increased. So they would, yes, there would be higher viral titers, higher pathology in the lung, and more cytokines in the macaques.
The interferon is not inhibited, right? Okay.
Exactly. Or I guess, yes, modulated.
Modulated. All right.
Yeah. And then there have been several studies as well looking at a specific subset of inflammatory monocytes in these in SARS-CoV-2 infected patients, so patients with mild to moderate COVID-19. So next, they performed flow cytometry on whole blood and on bronchial alveolar lavage from the rhesus macaques, and they did pre and post infection, and they saw that there was a lower frequency of these. This won't mean a lot to many people, but they're CD14 positive, CD16 positive monocytes specifically. So they had less of those that were treated when compared to the untreated macaques. So these inflammatory monocytes, there's less recruitment to the lungs, it seems like, and during this interferon modulation, which I thought was interesting.
Somehow the interferon is activating these and recruiting these monocytes, which themselves cause damage, right?
Exactly,
That's
yeah.
the idea.
And I think that maybe it's not only, like the interferon is obviously part of it, but then everything else that the implications of said interferons with the other innate cell recruitment, et cetera. Because then they also go into the next figure where they wanted to characterize the pathways that are affected by this interferon modulation in vivo, whether you do bulk RNA-seq of bronchial alveolar lavage, PBMCs, and whole blood. That was done after SARS-CoV-2 infection again. And they see that this treatment suppresses the expression of ISGs, so those interferon-stimulated genes, which you expect, but also inflammatory genes and neutrophil degranulation of the bronchial alveolar lavage of these rhesus macaques. Specifically, some of the genes, if we can, this is in figure 6, things like OAS, MX1, and then neutrophil degranulation, some specifically associated with that. So it seems like there are more innate cells that are being affected, not only the monocytes, by this interferon treatment.
So TWiV 900, burning down the mouse. We discuss why inflammasome activation in infected macrophages drives severe COVID. It's the same story, really, right?
Yeah. They also speak about how there is dampened as well inflammasome activation as well in the macaques, which I thought was interesting. But yeah, I wasn't going to go too into detail about that, but if anybody is interested, they do talk quite a bit about like the NLRP3 inflammasome and the AIM2 inflammasome. They look at that as well and see that it's also dampened in these interferon modulator treated macaques.
They actually do a nice job of, after each section, summarizing it, because not everybody does that. I was reading a paper for TWiM the other day, and I'm like, hey, why don't you summarize it for me? So here, interferon mod treatment in rhesus macaques was not only able to potently attenuate inflammation and inflammasome activation in the lower airway, but also systemically. So that's great. You don't have to look at any of the data. You can just get the bottom line if you want.
Exactly. Yeah, you don't have to go fishing for the findings. They're very clear about them.
I find these papers where they're looking at markers very difficult to read because the sentences are interspersed with CD38, CD69, CD274/PDL1, CD80. I mean, it just makes it really hard to read, right?
Yeah.
And your type 1, your system 1 wants to kick in and just skim through it. And then
or sometimes authors will set up, to test whether it was this or this. We did this set of experiments, and then the experiments get really complicated, and they just tell you the results. So at the end of the paragraph, then you really don't know which way it points.
It's good to have a summary. It is good to start with. We tested, we tried to address it. I like that too. It really helps.
Yes.
Yeah. And then they also did, so then this is after this bulk RNA-seq where they saw this suppression of the neutrophil degranulation genes, inflammatory genes, ISGs. Then to further characterize the effect of the interferon modulator within the lower airway, they did single cell RNA-seq on the caudal lung lobes that were obtained at necropsy. So these will be the lower lobes of the lungs of the rhesus macaques. Well, in this case, they obviously didn't say that they died from SARS-CoV-2. This must have been just euthanasia because there was no mention of them dying because of the infection. So, and then they see here, they show this UMAP, which was interesting. They're just showing the different macrophages and monocytes. Most of them 2 days post-infection were macrophages and monocytes. And they see again that the treatment effectively does suppress ISG expression, and similar to some of the things that we see in the bulk RNA-seq.
I love the name of the program they use to look at the single cell RNA-seq data. Surat.
Oh, Surat, yeah.
Isn't that great?
Surat, yeah. I guess I've heard that word a lot recently. So when I saw it, it was lost on me. But yeah, it is a funny word.
Most people have not, most listeners have not heard it in the
Yeah,
context of single cell analysis, but they look like Seurat paintings, right?
But it's spelled Seurat, Seurat.
Right.
Yeah.
It's the same as the artist.
The artist, yeah.
Exactly.
Yeah.
In case somebody thought it was like, I don't know.
Sir Rat.
SEW or Sir Rat. Yeah, exactly. Like Sir Rat. Yeah. So then in these lung lobes, they also see, as we were talking about before, from the single cell data, this dampening of the inflammasome activation and different cell death pathways during this SARS-CoV-2 infection. So I think that this paper is really interesting. I mean, a lot of people might see it and just say another SARS-CoV-2 paper, but I think it's just really interesting to go into more so like to better understand the virus, the interferon signaling, how that's affecting, especially within the first 2 days of infection, where it seems to be the most important. And overall, this study shows that whereas early in interferon 1 restrains SARS-CoV-2 replication, we know that this uncontrolled interferon 1 signaling does critically contribute to SARS-CoV-2 inflammation and pathogenesis in this model of moderate SARS-CoV-2 in non-human primates.
Yeah, it's important to note that it could be different in people, right?
Exactly. That's why in non-human primates, which once again, it's a mild to moderate, so we don't have the same pathogenesis that we do in humans, but I think we can still learn definitely a lot from this.
They say that the interferon mod achieved a Goldilocks effect of upregulating antiviral ISGs while reducing the expression of inflammatory mediators. That's good, right? Not too cold, not too hot.
That's right.
So the idea here is that if you have a good interferon response early in infection, it will control the infection, and then eventually it is cleared. But if you don't have a good response early, the infection amplifies, then the interferon tries to catch up, and you have an overproduction of ISGs and inflammatory, and then you go into that inflammatory phase, right? So the key is to really hit it early. So, and I think that's one of the
take-homes?
Best correlates of mild COVID is having a good early interferon and not having antibodies to your interferons, for example, right?
Yeah.
So having antibodies to interferon is a big risk factor for severe COVID. So it's really important to have an early response. And if you don't, I think, and they don't actually say this, but if you don't have a good early response, then the virus keeps multiplying, and then the interferon responds some more, and then you have this overreaction and inflammatory phase. So I don't know
that definitely contributes to this cytokine storm that we all talk about. That's definitely part of that as well from this interferon.
I mean, my view is that this interferon mod is helping us understand, right? But not particularly going to be a therapeutic in my view, right? Because they don't even mention
I think it's more so, exactly. I don't think it would be, they don't really talk about it being a therapeutic. I think it's more so just to understand the mechanisms of interferon signaling within SARS-CoV-2 infection. Yeah.
Very good.
I'm trying to think if there's a character or some other kind of name for. So the Goldilocks is that, it's just the right amount, but also the fact that early is good and later is not good to have too much interferon. I can't think of a thing for that, but anyway, So
when the horse is already out of the barn, you close the door, it's too late.
right.
Yeah.
Stuff like that.
No more interferon. Yeah.
All right. Our paper, also Science Immunology, I did think this is so cool. It's just not just the result, but the tech, the approach that they took. It's just really something different. It's an armed anti-immunoglobulin light chain nanobody protects mice against influenza A and B infections. And this is from Xin Liu, Thomas Ballinjand, Claire Carpenet, and Hide Pleu, who are at Harvard Medical School and University of Montpellier. And this is about using antibodies to to direct therapeutics at viruses. So everyone remembers that antibodies are made up of 4 protein chains, 2 heavy and 2 light chains. And in fact, most vertebrates have them, but camelids have them, but they also have heavy chain only antibodies, and they're called nanobodies, or variable domains of heavy chain only antibodies, VHHs. But I like nanobodies. They're small, and they're easy to get into tissues. They have a short half-life in circulation, but they don't have effector. They don't have that FC part to engage complement or to bind to FC receptors on cells. So what they did in this paper was to ask whether they could take a nanobody, which is directed against all kappa chains, kappa light chains. They took this nanobody and they linked it to an antiviral, zanamivir, which is a neuraminidase inhibitor. And then the zanamivir would direct the nanobody to either virus particles or virus-infected cells. And then the nanobody would bind to antibodies that are in the circulation, and those would have FCs, and then FC-mediated functions would eliminate the virus infection. That's the idea, right?
Right. It's too bad it's not open access, because figure 1A just really diagrams it well.
Yeah,
it's so nice.
Yeah. I was trying to do that because you know me, I'm a dinosaur, so I'm reading it on paper and I'm trying to think about what it looks like. And I was about to try and sketch it out, and I thought, oh, maybe they have a picture. And then they continue to have like that picture and other ones throughout as they do more experiments. And another name for the zanamivir is, or the drug sales name is Relenza. Right.
Yeah, so this is interesting. Relenza is one of the 2 neuraminidase inhibitors. You have Tamiflu and Relenza. So, classically Tamiflu is a pill you take. Relenza was originally a nasal spray, but apparently it can also be given intravenously. But it's not great. It's not a great drug.
And what I found is that it's a dry powder inhaled antiviral.
Correct.
So not even as a nasal spray. I mean, it's
a dry powder. That's correct.
It's a dry powder.
Yeah, it comes in a little canister that you have to spray. And if you have any kind of, airway reactiveness, it's not good for you at all. Yeah.
Right.
Like that would make anyone sneeze. It sounds like a dry airway.
It does. Yeah. But
also, before we get...
Sorry. Go ahead. No, go ahead.
I had like this little fact that I thought was cute. You were talking about how camelids have these nanobodies, and I just like to add, apart from camelids, well, also llamas, which are in camelids, but nurse sharks also have nanobodies, which I think is so interesting because they're so like phylogenetically distinct.
Only nurse sharks. Oh, cool.
Mm-hmm.
Oh, okay, because my diagram just says shark, but I have a diagram that I pull up for these occasions because I want to make sure I'm looking at the right part of the antibody molecules.
By the way, I have to take a digression here. Kathy, you said you like to read the paper, right?
On paper.
So I just subscribed to 2 magazines, The Atlantic and The New Yorker, right? So I have already 3 copies of The New Yorker here, and I've been reading them on the train, and I just feel so cool reading something. Like nobody else reads anything paper, and I really like throwback to the old days, and I love it. I flip through the pages. It's really cool. And then the other day I left mine on the train. Because that's a downside.
Hopefully somebody else is going to read it. Hopefully,
I just had this view that they're just going to throw it on the floor. It's like, what the hell is this paper thing? But because you're a lot less attentive with a paper than your phone or something. You're really careful to make sure you have your phone, but your paper. And I left it there, but I do have the online subscription. But I really enjoy reading the actual paper. It's cool.
Yeah, we actually, in our lab, we subscribe to Immunity and also to Science, and now we physically receive them. And it makes you, even papers that I wouldn't have wanted to read before necessarily, you're just flipping through and
That's the thing. The flipping through is what it's all about, right? You can discover things much. Now, people, ex-gens will tell us, no, you can search easily, blah, blah, blah. But I just flipping through.
It's not the same. Even the covers, we're obsessed with the covers now. And we always go, like, you see the cover, right? And we're all admiring the cover. And then you just open it into like the third page. You can read about the cover. It's just so much more easily. Of course, it's accessible online, but I feel like it's not the same. I agree, Vincent.
It's the same as an in-person bookstore or, going to Amazon and looking for your books.
Yeah.
I mean, even though they suggest, other people, you might like kind of thing, but it's not the same.
I mean, I still prefer books on a Kindle or something because it's lighter for travel. But the New Yorker is really, really light. It's just very thin and, it comes out like every week or something. So... Anyway, it's fun. So back to the paper. So these antivirals, the neuraminidase inhibitors, aren't very good. In fact, Daniel Griffin described a study a couple of weeks ago where the efficacy is really poor. You have to give it within 24 to 48 hours after infection, or it doesn't work. And we got a letter from Bob Krug. I don't know if you remember Bob Krug, Kathy. He's been retired for many years, but apparently he's listening, and he said, what about baloxavir? It's really good. One dose within 5 days will inhibit. So we read it. I read this letter, and I said to Dan, have you ever prescribed baloxavir? He said, actually, I have not. So we do. It's an endonuclease inhibitor. Apparently one dose given anywhere from day 0 to 5 will completely inhibit virus reproduction. Anyway, but we still need more antivirals, and that's one of the goals here. So let me give you the summary in case you want to go walk or run or whatever. A single dose of a nanobody, which is going to bind any kappa light chain. So the light chain of the antibody, it's called the kappa light chain. This nanobody will bind that. That nanobody is also conjugated to zanamivir. One dose protects mice from a lethal challenge with influenza A or B viruses, even when given a few days after infection, and no immunization. There's no vaccines involved here. All right, so I really love the coupling of the zanamivir to the nanobody, because they use two different technologies, and one of them they call the genetic approach, because they take the kappa light chain and they add 1, 2, 3, 4, 5, 6, 11 amino acids onto it, which are the target for an enzyme called sortase. The sortase is an enzyme normally involved in cell wall sorting reactions, where a cell surface protein is cleaved between a specific sequence. And so that's the sequence they put onto the kappa. And they also put this similar sequence at the end of, it gets cleaved and then attached to the zanamivir, which has another sequence, a similar sequence onto it. So the two of them are joined together. So then you end up with the kappa light chain, and you have zanamivir. You have a nanobody that's specific for the kappa light chain. Sorry, I misspoke. And that's hooked up to the zanamivir. That's what they call the genetic approach. And then there's going to be a second way in a moment that will tell you. So you attach zanamivir to the nanobody, and this has really high affinity for immunoglobulins. And in mice, over 90%, 95% of all the circulating immunoglobulins have the kappa light chain. There's another kind of light chain, but the kappa is 95%. And so this nanobody will now bind to those antibodies. And the idea is, it doesn't matter what those antibodies are directed against. What you want is the FC part, because the nanobody doesn't have its own FC part, so you're giving it one. And then the nanobody is going to be directed to the influenza virus or virus-infected cell by the zanamivir, which binds with very high affinity, right?
Right. So instead of having the combining site at the Y, the nanobody just connects to the zanamivir, and that's what directs the immunoglobulin to the specific virus-infected cell or virus.
The way they say this is very funny. We made use of sortase to install zanamivir at the C-terminus of the nanobody to make this adduct. I think install is a funny way to say that, right? I like it though.
Yeah.
All right. So now we have so let's say the nanobody, another name for the nanobody is VHH. So we have VHH kappa, zanamivir. It's an adduct. They're going to give this to mice and see if it protects them against influenza virus infection. And they're going to compare it to VHH kappa alone mixed with zanamivir. So not conjugated, just mixed together. And so they give it intraperitoneally and complete protection against lethal challenge when given an hour before infection. And the mixture of just the VHH kappa and the zanamivir that doesn't protect them at all.
Right. So said another way, when it's in the adduct form and everything's all connected,
right,
genetically,
it protects.
Then it protects. Yeah. But if you just put them in separately, they don't protect.
And also they do this with multiple strains of influenza. These are different isolates, I'm guessing, right? They do the Puerto Rico H1N1, 8. Yep. They do H3N2. They do multiple. It's not just with one virus.
And also influenza B viruses, right? So they all it works against all of them. And
this is really cool.
You can give this adduct at least 3 days after infection, and the animal's already having weight loss, still protects them from lethal disease. And so they say this is better than zanamivir alone because you got to do that within 48 hours, they say, for humans, or 24 hours for mice.
And it's interesting. It doesn't even give them a slight advantage. Like when you compare to PBS and giving it separately, the zanamivir with the VHH kappa, it's exactly the same. Like there's no advantage whatsoever. There's no survival. The survival curve looks exactly the same if you compare.
Yeah. And then they gave it prophylactically, so 7 days before infection, and challenged them on day 0. All the mice survived, no signs of weight loss. So it's sticking around for a long time, 7 days. Then the next experiment is pretty cool. They replace zanamivir. So zanamivir is basically bringing the nanobody and its attached antibody to the influenza virus infected cell or the influenza viruses. So they say, let's try another way. They, instead of attaching zanamivir onto the nanobody, they attach a nanobody against influenza virus HA. So again,
to a different surface protein of the influenza.
Right. So now, it's basically bringing the nanobody to a different surface protein. And this is a monoclonal against a conserved region on the stem of the hemagglutinin. And if you've been listening to us, you know that the stem varies less than the head of the hemagglutinin. So it's been a target for making universal influenza virus vaccines. This also protects against lethal challenge when given before infection with different strains as well. And you can also delay giving this 3 days after infection.
What's interesting is, did you notice that with the SD36, which is what Vincent's talking about now, they did almost 10 times the dose, though. They did 10 milligrams per kilo, as opposed to with the zanamivir, it was only, they started with 3 or with 1 milligram.
Yeah.
And then the max they went to was 5. I wonder why. I wonder if they did it with a lower dose and maybe it wasn't as effective. I'm not sure.
Yeah, I think they talk about the antibodies bigger than the zanamivir, so there may be some steric clashing going on, and so it's not as effective at bringing the nanobody there.
Okay.
What I found interesting is that they used click chemistry to put the HA nanobody onto the nanobody. And, click chemistry just got a Nobel Prize in 2022. And it's great. You go to the Nobel Prize site. The Royal Swedish Academy has decided to award a Nobel Prize in Chemistry to Carolyn Bertozzi, Morten Mildahl, and Barry Sharpless for the development of click chemistry and bioorthogonal chemistry. And then below that, it just says click, and the molecules are coupled together. That's why it's called click chemistry. They made this incredible system where you use a couple of chemical building blocks and they snap together, basically. And that's what they do here. I mean, you use specific chemical groups. And it's beautiful because you can make all kinds of molecules this way. And so they use that. They're really using a lot of cool tech in this paper. Mm-hmm. Anyway, so that's the influenza-specific nanobody. They also they installed a moiety into their nanobody, which allowed labeling with zirconium, so that they could see the molecules by PET scanning of the mice to see where the molecules are going.
Yeah, I had never heard of this. I had to Google it. When I saw the images, I was like, what?
Positron emission tomography, right? And they need to have an agent, and so they put a zirconium in. They put a moiety in the nanobody that will bind the zirconium. So now they can image it. Cool. And when you give this, now you give the VHH SD36 zirconium into uninfected mice, you can see that it goes to certain places like the heart and the liver. But in the influenza virus infected mice, you see really strong accumulation in the lungs, which it makes sense because there is where the influenza virus infected cells are. That's where the hemagglutinin is being produced. And so this nanobody is concentrating there. So in unaffected mice, it's going to a variety of places and it's concentrating in the lungs. So I thought that was pretty cool.
Yeah.
So this, so far, it seems these adducts seem to work, but how are they working? The idea was to put an FC region in. So is that how they're working, right? So they do this in cells in culture, MDCK cells, Madden-Darby canine kidney cells, commonly used to study influenza virus infection. And they infect them with influenza viruses. And they say, well, if we add this VHH zanamivir with or without antibodies and complement. So they could add all of them together, or they can leave things out and say, do you need complement? So they do what's called a complement. I forgot what the DC stands for. Complement deposition.
No,
complement-dependent cytotoxicity, right?
Right.
That's it.
Complement-dependent cytotoxicity, CDC. So there's another meaning of CDC, complement-dependent cytotoxicity. And they can show that you need all three in order to get cytotoxicity. You need to have the adduct, you need the IgG, the mouse IgGs that the nanobody is going to grab, and then the complement, which is going to bind to the FC receptor and then lyse the infected cells. Very cool. They also have an antibody-dependent cellular cytotoxicity assay. This is an assay where they're using T cells. T cells will have the FC receptor so that the antibody can bind to them and they can then activate killing. And they, again, show that you need the T cells, you need the polyclonal IgG, and you need the VHH zanamivir. So that tells you that this is working as they think it is. It's using the FC binding part of the antibodies that are bound by the nanobody. And then what I think is cool is they do this in mice. But now they say normally if you want to say that an FC part of an antibody is important for some activity. So here we think that it's important for the zanamivir adduct to kill virus-infected cells. You change the FC region. You change a few amino acids and it doesn't bind FC receptors any longer. And this is called the LALA mutation. I think it's because those are the amino acid changes that you make in the FC portion. But if you think about it, you can't do that in mice because you can't change all of the IgGs. There's more than one. You can't just change one gene and have all the FCs changed. So they say it's not possible for polyclonal IGs without germline engineering of mice for all IG constant regions. You can't do it. Well, they could, but it would probably take them 5 years to do it. Right? So instead, what they do is they make an adduct of zanamivir with a nanobody against albumin. Right? So these constructs will bind to albumin, which is in the circulation. It's in the blood, of course, and that will extend.
Yeah, it's the most abundant serum protein, right? Albumin. Yeah.
So they will, but they won't recruit polyclonal IgGs. So this construct, remember, the nanobody doesn't have an FC region. It's not directed against IgG. It's not directed against the kappa light chain as in the previous constructs, but now it's bound to albumin. This is such a clever way of answering the question: is the FC part really protective? So, VHH, kappa, zanamivir is protective in their experiment, but the albumin construct, you needed a 10-fold higher dose to get 100% survival, and if you didn't actually get weight, you still got weight loss. So it's not preventing infection as anywhere near as well as the other one. And if they increase the dose up to 30 milligrams per kilogram, which is the most they could put in, they still don't protect the mice. And 3 mg per kg of the nanobody conjugate did protect them. So it shows you that you need an FC interaction to get that high efficiency protection. I don't know what I guess the zanamivir is giving some protection in these experiments, right? That's why you're seeing that, but it's nowhere near as good as if you have an FC receptor brought in.
Yeah.
So so they also want to know how long these things last in the circulation, and they put in the zirconium labeled ones, and they can measure that. The VHH kappa and the albumin, you can find just a small amount in circulation at 6 days, but the nanobody, the anti-HA nanobody SD36, was cleared about an hour after injection. And
it's just surprising. That's so fast.
Very quickly, yeah.
Very fast, yeah.
I guess because of the FC is contributing to it, right? Because that's the difference with the albumin construction.
But even that, like an hour seems striking. But yeah.
They say the lack of recruitment of FC effector functions can be compensated for only in part by increasing the dose of the albumin zanamivir, indicating that a half-life extension does not optimally protect. So the albumin has a bigger half-life, but it still doesn't help in protecting the mice. And finally, another cool experiment. They say, so are immunoglobulins really needed for this protection? I say, okay, so let's take RAG1 knockout mice, which don't have B or T cells. They don't make antibodies, but they have normal amounts of complement, and they have cells with FC receptors like macrophages and natural killer cells. So they can take these mice and they can infect them with influenza virus, and then they can treat them first with their zanamivir nanobody adducts, and that doesn't protect them from influenza virus infection unless they give the mice polyclonal IgG as an injection. Because remember, the mice don't make their own IgG, so they can't supply that FC. But if you give them IgG, now the zanamivir adduct works.
It was a cool experiment.
I just think it's
really
great.
Cool.
It's so clever, right?
Yeah.
Yes.
So that's it. It's these adducts. And they say that we could probably, this is modular. We could adapt this to pretty much anything, any cell population that you could selectively target. You just somehow engineer the nanobody to be directed against those, and then the nanobody will pull in all these IgGs, which would potentially kill the cells, right?
So that's like, before I even got off of the first page, with the abstract and everything. So Vincent, you said, it's really cool technology and so forth. And I was thinking of it from the standpoint of, and can it be extended to other viruses or other microbes? And they finally get around to that in the discussion. But to me, it reminded me of this new section, or relatively new section in grant proposals, where when you talk about the significance, you should talk about the significance. But if you can extend it to beyond your virus and your system, like some side benefits, and this one would definitely have that. You could extend it. And they mentioned malaria, for instance, nanobodies against blood stage parasites, or viral pathogens that have envelope proteins like SARS-CoV-2, HIV, or Ebola, and so forth. So it just seems really like the sky's the limit kind of finding. I really liked it from that standpoint.
So I'm trying to figure out how you would target, let's say SARS-CoV-2 infected cells, right? So the nanobody, the key to the nanobody is that it binds the kappa light chains, right? And it brings in, but you also have to target the nanobody to, I don't know, spike.
The spike, right, for example.
So the antibody, though, is already directed against, so you conjugate the nanobody to, say, another nanobody directed against spike, right? That would be how, you have two nanobodies joined, one pulling in the IgG and one spike. Okay. You could do it to cancer cells, right? If you had an antigen on the cancer cell as well.
Mm-hmm.
Malaria.
So.
Yeah.
Yeah, like they say, blood stage parasites. Yeah.
Hey, right. We see no obvious obstacles to its translation. Really bold statement, right?
Yeah. Right. Yeah. And they also talk about how you could have a set of nanobodies against your intended target, but with each one recognizing a distinct pathogen epitope. So again, like maybe you could combine the hemagglutinin one and the neuraminidase one that they had here. Or, and they also say that there's real cost advantages to it. So yeah, low immunogenicity risk profile for nanobodies, and anti-human kappa light chains have been described. So yeah, it just seems remarkable with a lot of implications.
Apparently much easier to make than monoclonal antibodies.
Right.
You can make them in bacteria, in fact, because it's just one chain. And a number of people have said, monoclonals are hard to make and they're expensive. And so that's why, as the Omicron variants emerge, you don't see a immediate production of monoclonals because it takes time and it's hard to do. But nanobodies are not, I guess they are neutralizing, but not as good as monoclonals because the monoclonals have the two combining sites.
Kind of makes me wonder why it wasn't in Science. Instead, it was in the Science Immunology journal.
I think it's a cool finding. Yeah, it's really nice. 4 authors. Yeah.
Yeah.
Good stuff. All right. Let's do some letters here. Kathy, can you take that first one?
Okay, that is from Suzanne. Hi, Vincent. I've heard you comment recently on your numbers dropping, but I can't quit listening. I started listening in May 2020, and I haven't missed an episode since then because I'm completely hooked. I like everything about it. The weather updates at the start, learning the science, eavesdropping on your conversations about the science, and everyone's picks at the end. When I heard Alan comment at the end of episode 997 that TWiV recordings structure his week, I thought, listening to TWiV structures my week. I look forward to your release of your conversations with Dr. Daniel Griffin. I listen as I do house chores over the weekend, as well as the release of the regular TWiV episodes, perfect for walking my dogs. I will say that I greatly appreciate your recent push to do more explaining. I'd been feeling like I was understanding less in some of the recent conversations of non-SARS-CoV-2 topics, but the efforts by all of you to provide more explication has been a real boon. Thanks for all that you do, Suzanne.
Wow, a perfect listener, right?
Yeah.
Likes everything about it.
Yeah.
Thank you. That's just great. I wonder how many people like everything, right? If there are 60,000 listeners or 100,000, including YouTube, or more, 120,000. How many of those like it all, or how many of them like parts of it?
I'd say like 70-30, maybe 70% like it all. You think that's too high?
You think it's that much? That's a lot of people.
I mean, the thing with podcasts, it's easy. If you don't like it, you just fast forward. You just go, you just jump ahead. Like when I listen to podcasts at the beginning, if they have a whole bunch of sponsors, I just directly go to minute 7 and then that's it.
Yeah, you can do that.
I just go ahead.
Well, it turns out there are a lot of long podcasts out there.
Mm-hmm.
So people do that. And, remember people used to complain that our podcast was too long at 2 hours, but gee, there are a lot of other long ones out there and longer. Lex Friedman is 3-4 hours.
There's very long ones. Andrew Huberman, some of his podcasts are 4 hours long and he has like millions of listeners.
Yeah,
and some of them are 4 hours.
I just don't buy it's too long. Yeah. Sorry.
Right. Mm-hmm. And I remember when we suggested that you could fast forward through things, then we had the listener who said, I listen while I'm driving. I don't want to drive into a bridge abutment.
This is
so
true.
I always think of the listener driving into the bridge abutment because of
Couldn't
fast
you
forwarding.
give an oral command, Siri, fast forward 2 minutes or something?
Yeah, no, not yet.
All right. Angela, can you take the next one, please?
Yeah. Dave writes, thank you. Episode 997 of TWiV captures so much of what I've been seeing regarding viral evolution and the current direction of this pandemic. Thanks again for the straight up sober analysis of good questions that have been asked and what we are learning in the face of adverse conditions of a misplaced perception by some who may be well-meaning, and unfortunately, many who malignantly are not. Whatever, I'm present to tend that barrier. However, I might find a way. Thanks again, and keep up the good work, Dave. That was oddly worded.
It is.
Unless it was just me. It was awkward to read. But
I'm present to tend that barrier. Yeah. This 997 was Mohsen Saeed. Catherine writes, please share with Dr. Condit this episode, and Catherine provides a link to a podcast, specifically addresses how Dan uses Bach and creates his own music. I think he will like it, given his last pick of the week on TWiV. Okay, so what's this? Reinventing Bach. Switched on
It's
pop.
a podcast link, isn't it?
Yeah.
To Apple, yeah.
It's It's a podcast called Inventions/Reinventions by Dan Tepfer. And the episode is Reinventing Bach.
Yeah.
Oh, so
I remember that pick. It was really cool.
So Rich picked someone doing this, right?
Mm-hmm.
And here he's talking about it. Okay. If you ever learned classical piano, you probably tried to play one of Bach's inventions. The composer wrote 15 pieces containing the most important 15 keys in order to teach his son the fundamentals of piano and conservation. That's cool. Today, they remain some of the most popular pieces of piano music. Acclaimed jazz pianist Dan Tepfer recently revisited his childhood music books, seeing them in a way he'd never realized as a student. The inventions are much more than novice piano works. All right, cool. Back to you, Kathy.
And that is Justin. Even though we Americans dislike coins, I have a feeling you'll want one or many of these. And he sends a link, which, oh yeah, I remember I clicked on this before. American Innovation $1 coins, 2019 rolls and bags. So they show a microscope and polio vaccine, but not really with a icosahedron. It's a different polygon. Anyway, and then you may also and then they have, so that's the one from Pennsylvania. And then they have different ones from Delaware and New Jersey, and they have different images on them. So yeah, pretty cool. $1 coins about the polio vaccine.
Yeah, so Salk's vaccine was made at the University of Pittsburgh, right? So,
Yep.
And here there's one from Mississippi, the first human lung transplant.
Oh,
Is on that one.
Oh.
That's cool. Do I have one of these coins? I think I might have one. Someone may have given me one. I have to look at the incubator. Oh, I'm in the incubator. What are you doing? Hey, look, hold on.
he's going to go look on his shelf. He's looking now at all his cool things. I took pictures of it when I was there for TWiV 1000. I took pictures of his shelves.
Oh, really?
So I could look at my images and see if I find them. It's looking hard.
Ah, here it is.
Yep,
Oh, wow.
he's got it.
There it is. Someone must have given it to me. Here it is, the American
go.
Innovation, right? It comes
Yeah.
in this little case, and you slip it out, and then there's another thing inside that you have to open up, and it opens multiple ways, and there's a lot of text and images, and there's the coin. Yeah,
Very cool.
it's got the polio virus in a microscope. Yeah.
Yeah,
That is cool.
Oh, I wonder who gave me this.
you have it recorded somewhere else.
Probably. I mean, I certainly didn't buy this. Yeah, American Innovation. Neat. I knew it was underneath a book there. I knew I had one. So if you sent this to me, please let me know.
Email in.
Yeah, tell me if you're,
Vincent at micro.tv.
if you're still listening. Very good, Angela.
And then so that first link is just to that polio one. And then the second link, which I knew I hadn't looked at, is that just gives you all the ones. So that's where Vincent found the Mississippi one. There's an Indiana one, etc. So you can find one for your favorite state.
Do you want to read the quote?
I have to go back. Oh, yes. So it's on our next page. There's a cult of ignorance in the United States, and there always has been. The strain of anti-intellectualism has been a constant thread winding its way through our political and cultural life, nurtured by the false notion that democracy means that my ignorance is just as good as your knowledge. And that's a quote from Isaac Asimov from A Cult of Ignorance, and quoted Newsweek, January 21st, 1980, page 19. Well referenced. Wow.
Page 19. That's good.
Must have a paper copy of it.
Okay, Angela, you're next.
Okay. Rowan writes, "G'day from various parts of Australia. It's sunny outside my hospital room, but the only temperature I can tell you is in my internal of 37.3 degrees Celsius. I'm holed up in hospital waiting for a valve replacement due to a bacteria getting in and eating my aortic valve. At 34, this was a bit of a shock, but if all the various TWiV shows have helped me to take a second and go to the primary literature and read the current science to figure out my options. Early in episode 1031, Death on the West Nile, there was some discussion of the intersection of science and politics. There is a sense regularly expressed by people on the various shows that this is not political or politics, shouldn't be mixed with science. My opinion is this is seeding around that should be held. This is seeding ground that should be held. Science does lead to conclusions about political matters. Science of every stripe tells us that ivermectin is not an effective therapy for COVID, that vaccines cause adults, not autism, that the climate is entering crisis, and that choices we make politically do in fact have an effect on the world. If all scientists, as well-educated and informed people, abstain from the political discussion, then who is left to have it? Certainly not people who have spent years learning to put emotion on the side, to try to prove themselves wrong, or to iterate their views to more closely match reality. Instead, we get ideologues and grifters.
Grifters.
Grifters, sorry. Scientists need to be political. The world is literally on fire. And to be frank, I would love to be able to worry about whether my replacement heart valve will wear out in the next 34 years. But at this point, it seems moot. If we don't engage with politics, there won't be a healthcare system to fix my heart. Fight the next pandemic, provide basic care for pregnant people, or even just fight off the scammers doing psychic surgery. Robust engagement of the scientific community is a necessary but not sufficient part of getting politics to actually solve some of these problems. Without your voices and the voices of other scientists, there is no point people like me thinking about retirement. There is no point in people like me thinking about retirement. Love the shows. Please keep talking, and please be outraged. Rowan. That was...
Well,
very opinionated, Rowan.
I think it's great. Good luck with your valve. I hope you got it.
He was only 34. He was basically my age. That's so young.
So when politicians get the science wrong, yes, we do talk about it, right? We've been on Paul Offit's program. We've been talking about RFK Jr. for quite a few weeks now. But, we don't become politicians. We don't talk about just politics. So that's what we mean when we say we don't get political. But we do correct the science. Whenever a politician gets the science wrong, when a state says you don't need a childhood vaccine, when a state says you don't need to mask or whatever other things they say, we say that's wrong, right? But we don't say you shouldn't be gerrymandering, because that's politics, right? That's not what we do.
Exactly. It's when they put their noses in science and unfortunately are spreading misinformation that then needs to be clarified by scientists. Not that we're
so right now we're having these hearings in Congress about whether we should be doing certain kinds of experiments, right? Well, the Congress provides the money for science research, so they have a right, I suppose, to ask what's going on. But to think that it's unsafe and you have to have a hearing to talk about it and what kinds of experiments you're doing is just crazy. Like, well, why would they know that at all, right?
Well, and they're not qualified to even understand what that means, right?
Yeah, they're not. But if you say that, then people say you're being elitist, right? So...
No, but I mean, okay, then I'm elitist, I guess.
I'm just telling you what people tell me all the time, all the emails and comments that I see, right? But
no, you yourself, for example, you have 50 years of experience almost. That means something. That's...
to some people it doesn't, though, right? It doesn't. They can have an opinion that they think is of more value. But yeah, I understand. The politicians don't understand the science, no. But what they're doing is putting on a show to get votes, right? And that's what bothers me most. If you do something that puts people's lives at risk just to get votes, this is totally unacceptable, right? When Florida and Texas make regulations that you don't have to get this vaccine or you don't have to wear face masks or whatever. It's swapping lives for votes, and I think that's unacceptable. So to that extent, we do venture into politics, but only to fix the science. Does that make sense? Yeah?
Mm-hmm.
Yeah.
Kathy, you're next.
Matt writes, RFK show rebuttal, reaching the 30%? Dear TWiV, if 30% are, quote, not liking, perhaps they have not been reached. What about taking some of their comments and writing specific responses? This could not be true because. That source was discredited because. Kind of like a fact-check resource. I listen to you. Makes sense. They rebut. I still don't know where the flaw is in many of the RFK points. I would love to see an RFK rebuttal page, or would that be the equivalent of a debate on paper? I usually don't remember the TWiV points when I'm listening to the RFK point. I would like to know the statistic source for 90% of COVID deaths among the among the unvaccinated. I told that to a friend. She shared what her open-minded doctor said, that he was terrified for having taken the vaccine. He has seen in the hospital many vaccinated patients who had multiple recurrences of COVID before finally dying of complications from COVID. My friend is open-minded too, but we feel such a dearth of helpful information on the grassroots level. Thank you. And this has a different signer than Matt writes, so maybe it was written by May, or was it written by Matt?
Oh,
Anyway.
I have to fix that. Yeah.
Yeah, whoever it was. Well, if it was written by May, then May has a master's in science education.
Yeah. So the number, by the way, Kathy, is 30%, because I mentioned this last time on the RFK episode we did with Dan Wilson.
Mm-hmm.
It had 30% dislikes, which is way higher than most episodes. Most episodes have 5% dislikes, and that one is 30%. So,
wow.
That's what she's writing about.
But do you think that's because people that wouldn't normally listen to TWiV, because we also had more views in general. So it's not necessarily that TWiV listeners are disliking, but that other listeners came.
I agree. Yeah. I definitely think that it attracted outside of our usual. Right.
At least they listened. Hopefully they listened.
Well, so some kind of rebuttal page. I don't know. Would Dan somehow, Dan or Dan plus you or something, Vincent, put something together, kind of rebuttal page?
I'm happy to do it. I'll ask Dan. Maybe some people respond better to seeing it on paper, because I'm
kind of like an FAQ. Like,
yes, absolutely.
Why isn't this statement correct?
Because I'm kind of sad that May says I still don't know where the flaw is in many of the RFK points. Right. Yeah. I think we did go through it, and certainly Offit has gone through it in his Beyond the Noise, but maybe it doesn't stick when it's just spoken for many people. I can understand that. So
yeah, the one that I've told over and over again is the fact that, first of all, vaccines don't have mercury in them anymore, and that the mercury that is really dangerous is the methyl mercury, and that mercury that was in vaccines was ethyl mercury, but that RFK says there's only one mercury on the periodic table.
Yes.
Well, what's the flaw there? The flaw is that he doesn't understand chemistry. And so as I was talking to my neighbor this morning, it was like, okay, there's only one atom different between hydrogen peroxide and water, H2O2 and H2O, but they're very different. So methyl mercury and ethyl mercury are very different chemically. It's not about the periodic table.
No. Oh, so his claim that no vaccine has been placebo controlled is just
it's
wrong.
Just simply false.
It's wrong.
It's just wrong. Yeah, it's just wrong.
Yeah.
So
there's two for you, May.
Anyway, I think I don't mind a fact. I don't mind doing it. I'll see if Dan wants to collaborate.
Yeah.
This statement now, let me talk about this, 90% of COVID deaths among the unvaccinated. So both Rich and Daniel Griffin have both said that in the last few weeks. So on the last update, I asked Daniel, the one that's going to drop on Saturday, which has already dropped if you're listening to this.
Right.
He said, we have a bunch of people in hospital today who are there for COVID, not with COVID. So they came in because they're sick with COVID. I said, so these people who are hospitalized, are they vaccinated? He said, yes, they're all vaccinated. So this statement is not correct. He said, what is correct is that they're all over 65 and they have comorbidities and they waited too long to take an antiviral, right? It's because they said, I didn't feel so bad. Let's wait and see. And their doctors agreed. And then they end up progressing and it's too late to take an antiviral. So they're all vaccinated. You get infected. The Omicron, the latest Omicron is immune evasive. So you're going to get infected. And if you're old and have comorbidities, you can progress to severe disease unless you take an antiviral. So that's the story. They're mostly in elderly people with comorbidities. They're all vaccinated, he said. So, May, that statement is wrong. Sorry about that. And all the other people who have been saying, where are the data for that? It's not correct, yeah. All right, let's see here. One more here. Actually, we can finish them all. Yeah, let's finish. Jeannie writes, hello, retired rural and remote RN living in Canada's high Arctic here. The quotes relate to the absurdity, the quotes around retired. The absurdity of thinking I'm retired as long as I still have my license, as I'm regularly doing casual contracts at the health centers of the indigenous communities of the Beaufort Delta. At any rate, your discussion of whether or not plants are alive fits nicely with an idea that has been kicking around in my head ever since I became aware of the concept of self-generating complex molecules around deep sea vents, which is hypothesized to be the precursor to life. The idea is this: maybe the idea that there is a difference between these early complex molecules and molecules that are considered distinctly alive is incorrect. Maybe the sensation we all have that there is an essence about living things that makes us conscious, aware, even possessed of a unique and eternal spiritual essence, in at least or human form of life, others, is a function of chemical and electrical impulses in our brains, and the best explanations our brains have yet to come up with to give meaning to these impulses is that we are alive and somehow different from things that aren't alive. It occurs to me that all the things that living things do that make us call them alive are entirely possible for a complex molecule that isn't alive, and that, in fact, we are simply complex molecules with varying degrees of complexity, but nothing is more alive than the next thing. That we evolved neurobiology that allows us to believe we are alive is a happy but not necessarily essential accident. That would be consciousness, right? Having a nervous system. I hope this doesn't seem too dark. In my neurodivergent brain, it just makes sense as a possibility and doesn't really frighten me, as my ability to believe I'm alive is not going anywhere and is just as powerful, even if it's simply an illusion of chemistry and electricity. Happy to be shown how my theory doesn't hold up. I think this is lovely. I love that last part, is not, even if it's simply an illusion of chemistry, I'm still going to be alive, so I'm not worried about it. It's great.
Yeah, that's good.
So the molecules, well, the problem is, Jeannie, and Jeannie is the knitting crone. Oh, cool. We make up the definitions of what is alive and what is conscious and so forth. So you could have your own definition. I don't see what's wrong with that. Are the molecules alive that are able to self-reproduce? I think, for me, it would be a stretch, but I have no problem with your logic. I understand what you're thinking. You guys have any thoughts about that?
Yeah, I agree.
Yeah.
Because we got into this discussion the last time, I think, because of someone, I think it was Alan's pick about how the Buddhists view this, right?
You talked about bacteria, right? How bacteria were conscious, or they were considered.
Yeah. Where do you get? How far down do you get consciousness, right? And
yeah,
because you see a bacteria reacting to a chemical gradient, and well, it's just a chemical thing, but maybe that's part of it. I don't know.
Yeah.
I think you have to be open-minded, and I think thinking about it is really important, right?
Mm-hmm.
I would think like the consciousness would be linked to a more developed central nervous system is how I would think of consciousness, more than like a bacteria or something. But that's just my own personal.
Well, I think so.
Like I've thought about it in animals a lot, like studying, like I thought about it a lot how a mammal has like a more developed nervous system, or a vertebrate versus an invertebrate. And if an invertebrate can be considered conscious, I've thought of that a lot, but it's hard to define, right?
Well,
like we don't know.
So you said something interesting, a more developed nervous system. So you could say that the bacteria reacting is a kind of it's a primitive nervous system, right?
Exactly, but it would be like extremely primitive, as opposed to,
sure, of course,
like the mammalian nervous system, let's say, or any vertebrate really. But yeah, difficult.
I think
to
this
answer.
is a very interesting discussion, and is a subject for a whole show on its own with people who think about this all the time.
A whole podcast about consciousness.
Yes, there probably are. Okay,
sure.
Back to Kathy. We have two more. Yeah.
Erica writes, hello, Vincent et al. I came for the COVID science. I stayed for the awesome. Though I've no medical background, I enjoy the show so much that I once had aspirations of going back and listening to all the TWiVs. Though it quickly became obvious this is a task well beyond my time constraints, I still queue up an old episode when I have the chance of it. And because of this, I'm in position to offer an alternative point of attribution on a quote that has come up a couple of times lately. I chickened out of saying anything when it happened before, but I would be a terrible TWiV family member if I let it go a second time. In episode 1026, Debunking JFK Jr., Kathy mentioned a quote from one Neil deGrasse Tyson: "You can't use reason to convince anyone out of an argument that they didn't use reason to get into." As it happens on TWiV 14 from January 5th, 2009, in relation to HIV deniers and malignant skeptics in general, Alan said, "You're not going to be able to reason themselves out of a position they didn't reason themselves into." Now that I see the quotes side by side, it's even more remarkable how strikingly similar they are. Of course, I don't know that Alan was the first person to say this. Maybe they both picked it up from some other source for which I don't have a reference. But he does have a full decade on Neil, and I just think it would be so great to hear you all attribute this concept to one of your own. Yeah, maybe more people in general know Neil's name, but for my part, and I imagine yours too, I would take Dr. Dove over Dr. Tyson any day of the week. In any case, you are all amazing, and thank you so, so much, Erica.
It's lovely.
Well, thanks, Erica. That's fun to know.
That's a lovely letter. It could be that, well, Tyson was around in January 2009, so maybe Alan heard it from him. But we'll have to ask Alan where he heard it from.
Right. Or, you know, it might be in Bartlett's quotations, but
yeah,
it is a great quote, though.
Yeah.
It is
yes.
Yeah.
Oh, two wonderful fans today here praising TWiV.
Several. Yeah.
That's great. All right. Angela, take that last one, please.
The last one is funny. Beth writes, "Come on. I was listening to TWiV 1031, preparing my dinner of chicken and roasted cabbage. As I was sitting down to eat, you began talking about the virus on the cabbage. Oh no, imagine my relief hearing it's harmless."
That's wonderful.
Yep.
Very nice. Okay, time for some picks of the week. Angela, what do you have for us?
Yeah, so I just got back from vacation. I was in the Canary Islands for a month, and I'm a free diver myself. I love I'm basically anything aquatic in the ocean. And this new documentary came out on Netflix, which is about freediving specifically. It's called The Deepest Breath. And it's amazing. It's about Alessia Tecchini, sorry. She's a world famous freediver. And it's about like her story and her path towards like reaching the deepest depths and like all of the things that she overcomes in doing so. And it's just really moving, and it's a beautiful story. And it's a real story. So it's her in the documentary. And it kind of gives you like a feeling of the world of freediving and how it's not kind of, people think of it in a certain way, but it's not necessarily like how people think. It has to do with a lot about like mental clarity and like calming your mind and staying really like relaxed, as opposed to when people think about it, they think about people swimming down really fast, which basically freediving is not like scuba diving. So you don't have a tank on your back. You're just going down and up with one breath. And this woman, she reached the world record at 104 meters, which is 312 feet. And that's going down 312 feet and then also coming up all on one breath. And the amount of pressure that deep, like myself, I've been down, my max depth is 26 meters. So it's like just over 75 feet. And you can already feel it so much in your lungs. I can't even imagine like 104 meters. It's crazy. And it's just it's a really cool documentary. Anyone that's interested in diving and in the ocean. And even if you're not, you will be after watching it.
So go ahead.
I was going to say, so Angela sent us all some pictures and this video from her vacation. And later, I saw that there were comments below about the photos and the video, but I started watching them first. So I started watching this video. First of all, there's this amazing wetsuit that Angela's wearing. It's all patterned. It's not boring black. Oh my gosh.
It's a whale shark. Yeah. Whale shark pattern.
Really cool.
Yeah.
And then, then I'm just watching it and she's going deeper and deeper and longer. And I'm starting to realize that this is all freediving. There's no tanks. Oh my gosh. And then she swims through this tube And then the photographer behind her swims through the tube. And I was just like, ah, I was getting really nervous. And obviously she sent the video, and then I read the explanation and everything, but I was a little nervous in the middle of it, but it's very
So
cool.
how much time is involved from top back up?
It depends on the dive, but like that video was like 1:20 or something like that.
Okay,
But it also depends, like when you do static apnea, like when you're holding your breath static, or dynamic. Like static, I've done like 4:36, but dynamic, it's only ever max 2 minutes, right? Because when you're moving, your muscles use oxygen. So it's always less, which is why we do like really slow movements. Because if you start like jerking around, you'll use up a lot more oxygen. So which is why like when you see in that video, everything is really slow and calm. Like you always have to maintain like calm. And obviously going through like tubes or like little things like that, you can't be claustrophobic, and you have to be like mentally very clear. If not, if you get nervous inside, obviously you're like, at that case, I was like 40 feet down. So if something happened, but it was all good. I had a safety diver with me and somebody else on the surface, which is very important. If you watch this documentary, you'll see you never go free diving by yourself, because you can have like a blackout, a shallow water blackout. And if there's nobody there to save you and to bring you to the surface, you'll drown.
So you should have someone with a tank on with you. Right?
No, not with a tank. It should always be a free diver.
Okay.
Because if you black out, let's say at 10 meters depth, like 30 feet, you never want to give that person oxygen, because the capacity, so like Boyle's law, because gases shrink, right? So whatever air you take from the surface, if you try to take in air at like 30 feet, let's say, and then try to ascend towards the surface, your lungs can expand, and you can actually like cause damage to your lungs. So you never actually inhale. So you have safety free divers that can dive however deep you're going. They dive that deep in case something happens. So.
And then a question about the breathing. So when you were talking about static, and the other word was dynamic.
Dynamic. Yeah, just when you're swimming is dynamic.
Right. Okay. But is part of it, do you get some seconds because you're exhaling at the end? Or, I mean, I never see bubbles.
No, so you never exhale. So you always, it's important that when freediving, you inhale like really deep into the caudal parts of your lungs. There's like specific breathing that you learn to maximize your lung capacity. And then you hold on to that breath until you reach the surface again. And you only exhale once your face is out of the water, because that's the oxygen you need, right? Like if you don't have oxygen, you can't go deeper. And a lot of people, like when we snorkel as kids, we always like exhale and go up and down because we're only going for a few seconds, but every molecule of oxygen is needed, right? So we keep all of that in, and then and then as you go down, though, once you pass like 30 feet, maybe 40 feet, depending on like how much fat you have on your body and how much weight you have, you start free falling. Like you don't need to actively swim as much, because your lung capacity decreases, right? So like you start descending, and your lung capacity, those molecules of nitrogen, oxygen, they're all getting closer together. Like the gases are getting tighter, because for every 10 meters depth, you have one more atmospheric pressure. So let's say like on the surface is 1 atmosphere, right? And then at 30 feet, you have 2 atmospheres. And then at 60 feet, you have 3 atmospheres. So your lung capacity also, your lungs they're shrinking, right? So if the air was like this big, well, then at 60 feet, it'll be like this big. So then you also sink faster because you're not as buoyant. So you hold your breath though. And then as you come up, you feel the pressure like expand, like you feel your lungs expanding again as you're surfacing.
Does it hurt as the lungs are expanding?
No. You can feel like a slight, when you go down, you can feel the pressure. And in the beginning, if you're not used to it, it can be kind of overwhelming, because you feel like, because what you do is you have to do something called compensation, where as you feel your inner ear feels like there's pressure in it, and you need to equalize it using your throat. So you just kind of like pinch your nose and you blow out, and you can feel your inner ear pop, because there's a connection between your middle ear and your throat. But then once you pass like 60 feet, that gets harder to do because of the air in your throat, and like everything is getting more compressed. So it gets a little bit more difficult, and you can feel that like tight pressure, but you get used to it. And
do you have the urge to breathe out? Do you have to consciously work against such an urge, or does that urge go away?
So to exhale, not so much, but you have an urge to breathe for sure. So what we call we call them diaphragmatic contractions, where you feel, some people feel it in their throat and some people feel it in their diaphragm, where you hold your breath for so long, you feel like that urge to breathe. But when you're free diving, you have to, the main thing that you learn is like, once you pass that urge to breathe, it's very uncomfortable. But then once you pass that, you have like another 30 seconds, or depending on the person, it's everybody's different, but you could have another like 30 to 40 seconds before you get another urge to breathe. And it's uncomfortable. But then when you pass that, you can get like another 30 or 40 seconds. And for me, for example, I feel it in my throat, and all I do is I kind of like close my eyes, I concentrate, and I swallow. I try to swallow, and then it passes, and then I can continue diving. So
Wow.
Okay. So this is something that practice helps, right? You get better with practice.
Oh yeah. Lots of practice. And please don't do it if you go with somebody professional. There's you can do professional certification for free diving. Please don't try to go diving deep without like any supervision, because it can be very dangerous. So you can get like basic free diving course, and then there's different levels, like level 1, level 2, level 3. And yes.
No, thanks. I don't think so.
This week in free diving.
So how many years have you been doing this, Angela?
Like 6 or 7. I've been free diving more because I used to work at a diving center, but like with tanks. Like I've done scuba diving for like 15 years, and I've always dabbled in free diving, but a little bit more. And I'm not even like, I'm nowhere near pro, right? I'm very just like, I do it for fun, but I'm safe, obviously, but I know things, but I'm not like a professional free diver. Like if you go watch this documentary, you'll see that these people are amazing. I'm just like,
so one more thing, you had really long fins, I noticed, right?
Exactly. Yeah. The fins are much longer than scuba diving fins to displace more water. So that way you can go slower and move farther, as opposed to with like a shorter fin, you'd have to move your legs really quickly, which would also make your muscles contract more and you would use more oxygen.
Yeah. They look very cool, I must say. Those long fins.
Yeah, they're cool. Yeah.
All right. Wow, that's amazing stuff. Kathy, what do you have for us?
I have something not nearly so exotic or maybe scary sounding, but it's about pink snow. And my friend and TWiV listener, Mary, sent it. And it's a really cool article written by a very good science writer for the Seattle Times. And this writer has also written books that Mary recommended. But it's about algal blooms that cause pink snow and how there's possibility that they could accelerate melting. And it's about the scientists who are investigating this and how they take their samples. And so there's lots of cool samples of them with basically 50 mil conical tubes collecting their samples, kneeling in the snow and so forth. And it's just really well written and really informative. And they basically don't have enough data yet to say whether the increased algal blooms are leading to more snow melting, because in fact, or that it's related in a direct way to global warming, because it does say somewhere that if there's too much heat, it's harmful to the algae. So again, it might be some kind of Goldilocks thing, but it's also called watermelon snow in some places.
So cute.
And it's just a really good story. And then Mary sent me a link to a book that I'm going to try and get and read too about a different topic. But she just highly recommends this science writer, and I do too. It's kind of a nice story and about some women scientists and watermelon snow. So take a look.
They have 50 ml conicals.
Yep.
It's so cool. Back in the lab.
Yeah. They don't have Kim wipes out there on the ice, or at least that we can see, but it's cool. Definitely lab science.
There's one nice photo
really
of
cool.
Someone handing a bag of 50 mLs to, and it's so colorful because their clothes are different. One of them has blue gloves. It's neat. Very pretty picture.
Yeah.
My pick is a short video that we made called Scientists versus PowerPoint. So a couple of, months ago, we had a symposium in Maryland for the retirement of Kostya Chumakov, who was at the FDA for many years, and a bunch of speakers. And I used my laptop and we had to interface with Zoom and film everything, and it was a little complicated. So a lot of people had trouble with the PowerPoint. And so Chris Kondayan, who recorded it, said, we have to make a blooper video. And so this is it. It's really funny.
Cool.
It's very funny about what happened to everybody. Cool. And if you know some of the participants, you'll find it. Their reactions are really funny. It really is really good.
Oh, great.
We have a listener pick from Fernando. In the last few pods, you riffed on manipulative anti-science argumentation and on cognitive bugs, and most recently on your pick of thinking fast and slow. I'd like to complement those comments with 2 book picks that, in my view, offer much more nuanced evolutionary views of these questions, focused on our evolution as social animals and on language as a technology for persuasion and maintenance of social standing. In my opinion, they supplant thinking fast and slow while paying proper attention to the data that the book is based on. Here they are: The Enigma of Reason by Hugo Mercier and Dan Sperber, Language versus Reality, Why Language is Good for Lawyers and Bad for Scientists by Nick Enfield. The subtitle of Language versus Reality is especially apropos to the debating question. Yeah, it's good. Thanks again to the whole TWiV crew for teaching us and making us think through entertaining argumentation. Thank you, Fernando. I have to look into these because I'd love to see the data on which thinking fast and slow is based on. Yeah, it's really good. By the way, I'm still reading Thinking Fast and Slow, and he periodically gives more problems, and I'm really happy to say that now I get them. I don't get the wrong answer. I take my time and I think about it. I was on the train the other day and he had a question, and with like 5 minutes, I'm saying, okay, I think this is the answer now. Is this right? And I went through all different ways of checking it, and then it was right. So if you spend a little time, you can usually figure things out. Thank you, Fernando.
I wanted to point out what you mentioned, the Chumakov symposium or whatever it was, but I really enjoyed that episode, and a couple other people have said that to me, and I've also run into a couple people who have said, sorry, Kathy, I haven't been listening to TWiV much since the pandemic, but if you know of really good episodes, so I've pointed them toward that one. I really like that one.
Yeah, the title Chumakov Viridi, because his parents
perfect.
And him were all virologists. So
right.
A family of Chumakov virologists. Perfect.
Yes.
Yeah, it was a very interesting story. Yeah.
Yeah.
That's TWiV 1033. You can find the show notes at microbe.tv/TWiV. You can send a question, a comment, a pick. We like to hear what you like and even what you don't like. It's all good. TWiV@microbe.tv. And if you enjoy our work, we'd love to have your support to help keep this incubator incubating. You can go to microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Angela Mingarelli is at McGill University in Montreal. Thank you, Angela.
Thank you so much. Today was great.
When are you going to go on your next vacation and do some more freediving? Next year, I guess, right?
Next summer. If I can fit it in before then, I will, but I don't think so next summer.
So I've never been to the Canaries. Are they really nice?
They're beautiful. And there's direct flights now from New York to Tenerife with United and with Air Canada. So you should check them out. They're actually pretty cheap too.
Yeah, Angela had a layover here in New York. So she came into the incubator and we went to dinner. It was fun.
Oh, nice.
Yeah, it was really nice. Yeah.
Yeah, we did take a picture. I have to show it to you, Kathy. Right. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.